We're the industrial side, right? We're the optimists who have to basically take that star field that you see and say, this one will work. And we're going to show you how we begin to do that. A couple disclaimers. One is that the title changed. The reason the title changed is because my feeling was to a swine group. First of all, the first disclaimer is I'm not a vet, and you just heard that. So that was number one against me. Number two against me was if I tell them when we talk about poultry, how interested are they going to be? Are we going to have an exodus to the door as I start to talk about poultry? But we're further along in poultry. So we're about 2.5 to 3 years in on poultry research, understanding early gastrointestinal colonization and what that can mean for performance. And then I'll kind of finish with the work that we've only recently started with our partners at NutriQuest, who actually, Musser, is he here? He was the one who had the idea for the presentation. So I tried to ignore his email several times, but it just kept coming. So, but I do appreciate the opportunity to be here and talk about this. Oh, and the last disclaimer, this is just as our facility here at Agro BioSciences. We're in the Technology Innovation Building in Milwaukee County Research Park. It's a blue sunny day, like all days in Wisconsin, except when they lose to the Vikings. So, a stab. All right, so we want to talk about the role of colonizing bacteria, and in particular, these are the early colonizing bacteria and what they can do. And in blue, I've got 4 major headings that we want to talk about. One is that they're important in immune development and immune function. The early colonizing bacteria play a critical role in developing both the acquired and the innate immune system. And this is something that we learned about 10 to 15 years ago now. So it's really understood, and it's really seen a huge renaissance in terms of understanding the gut microbiome and what we can actually do in human world and health. In addition to that, and this is where I have to narrow down, because in terms of opportunities for companies, what we have to work with is a GRAS list. So generally recognized as safe microorganisms. So rather than look at that list that Heather showed you that was the predominant 16S analysis of the core microbiome, I want to ignore that. I have to ignore that. Because what we have to actually look at is things we can actually grow and feed. But critical enough is the fact that the lactic acid bacteria on the epithelial surface are known to do a number of things that are important for development of the immune system in that young animal. First of all, they can process antigens. Second of all, they can call immune cells to the GI tissue. And then more importantly, I think the work we did a few years ago was actually show that they bind to toll-like receptors and play an important role in quiescence or control the inflammatory response in the pig. In addition to the immune development and function, one of the things everybody hears about is competitive exclusion, and really in a bigger sense, that's really about homeostasis. And so the microbiota, and particularly lactic acid bacteria, play an important role where they actually can maintain normal gut function and gut health and barrier function by communicating back and forth with the host. And the work that's coming up now is indicating that microRNA from the host actually communicates with the bacteria, and that certain key organisms are either upregulated or downregulated based on that microRNA signal from the host. The last area, which I think is pretty interesting, and one that probably a lot of people haven't thought about, is that simply enough, microbiota process complex feed ingredients to make them more easily absorbed. But more importantly, what can we do to the phytochemicals, the polyphenols that are in the diet every day? Can we get the nutrition and benefit from those if we have the right microbiota to further process those and make them more available? And so we also work in the area of bioavailability of polyphenols, which are an important part of the diet. So, simply enough, our objectives for this study, and this was the one that we started in poultry, was to actually isolate and characterize the early gastrointestinal communities and identify if we could find a core microbiome. Another disclaimer, microbiome, microbiota. So microbiota is the bugs, and microbiome is the collection of the genomes. In this case, I'm not going to show you the detailed microbiome, but I'm going to be showing you the microbiota level. So our work was then to identify the core microbiome based on meta-analysis, the meta-sequencing work that we did to characterize the actual key isolates. And then, as Heather also pointed out, Just because you're there doesn't mean you're important. To understand functionally what those bugs did, and we'll show you some of that work, and then show you the commercial side of it. When we feed them, do we see an effect? So, one of the things about poultry systems that you probably don't think about is quite a bit different because it's the only livestock system in which the offspring is completely separated from the maternal line. And in fact, the things that they do to actually remove the pathogens from the egg to prevent vertical transmission is an important part of the story of what we need to dig into. As they use sanitizers, inject antibiotics, and do the rest to actually, what they are concerned most typically of is a vertical transmission of pathogens, what they actually set up is a dysbiosis within the system, because you've also killed the normal microbiota that were going to be part of that gastrointestinal tract of the hatched bird. So, our study began with that concept that we probably have a system that is creating the dysbiosis that we're seeing. And dysbiosis, by the way, leads to problems of avian pathogen E. coli in early stages, high levels of mortality at day 7, and dysfunctional performance in many, many flocks. So our job was actually to look at day of hatched birds, and we did this by collecting 10 birds per flock, and we also collected the hens representative of those flocks. And those, in this case, hens are pretty expensive. They don't only have 10, we can only have 6. And we do that for at least multiple complexes for each of the companies. And now I think we're up to our 10th company. So we're actually doing that to actually look at, then, the entire GI tract, doing it both the independent cultural methods as well as cultural methods. On the cultural method side, because of our early work, we're able to understand that it was E. coli as well as lactic acid bacteria that were critical in the early stages. So therefore, we use 2 different types of media, and we'll show you some of that data quickly. In addition to that, we'll actually show you the high-level work that we're doing at TRFLP, so terminal restriction, fragment length polymorphism, which kind of is the 10,000 feet, according to what, seeing the details. Once we understand where the differences are, then we can begin to understand where we might want to do deep sequencing, and we deep dive using Illumina sequencing today. So we'll kind of go through this data as we go. Here's some of the microbiology quickly. Hen microbiology on the first side, just looking here at lactic acid bacteria, or more importantly, these are lactic acid bacteria on selected media, breeder flocks, and you can see all birds had detectable levels. In fact, 16 of 18 birds had populations that are above 1 million. Representative birds, they have hatched birds from that same flock. You can see 35 out of the 60 birds had populations that were quite high. All birds had detectable levels, and you can see the numbers. So that's complex 1, company 1. Here's company 1, complex 2, in which we actually see similar data in terms of the ones we actually plated. High levels of populations greater than 10 to the 6th. Now the shocker. Lactic acid bacteria populations from day of hatched birds from the identical flocks, in which case we see that only 12 of the 60 birds, or 20% of the birds, had detectable levels of lactic acid bacteria at all. And so what we have here is a naive gut. And I'm going to show you the data relative to E. coli here in just a minute. So here's complex 1. Complex 1, representative E. coli, avian pathogenic E. coli in the hens, and representative avian pathogenic E. coli in the day of hatch. So that bird is far from sterile in the GI tract. In fact, you can argue at the 10 to the 6th, 10 to the 7th levels, you probably have a near septically infected herd that's being hatched. So this is the kind of concern that you have. Representatively, our sterile complex, complex 2, they were so good and effective, and I couldn't tell you all the things they were doing because it's part of the proprietary relationship we have with them, but they were so good at preventing the transfer of microbiota from the hens and at the eggs to the actual setting chambers and day of hatched birds that we only had 1 sample that had E. coli. Remember, most of these were actually had no lactic acid bacteria, and we don't even get sequence amplification when we do PCR. So basically nothing is there. By the way, we had good and poor performing right there in front of your eyes. The good performing was the complex system that we saw. This was the prior one. Complex 2 was actually the poor performing. So we dig in and look at the rabbits in sequence. I'll kind kind of jump around here, but it's going to move it pretty quick. I want you to just see the pattern you see. Excuse me. So the pattern you should see is that these are representative birds from the flock. These are the day of hatch birds, and right about here becomes the hens. And so you can see the pattern here looks more like a shotgun. And so this is actually indicating the diversity within the hens that is not represented within the day of hatch birds. And so isolates within each bird were often identical. You can see it's a run. It's a run of the isolates that almost are all identical in this case. This, by the way, is in groups of 5. So you can see the runs are almost perfect sets of 5. Boom, boom, boom. Those runs basically tell us that the lactic acid bacteria from those birds, which were the predominant lactic acid bacteria that we isolated, were in fact nearly identical. Isolates, in fact, within the same flock are often not identical, and then we did see some idea or evidence of a core microbiome. Now, core, as we said, is relative. This would be relative to this group of birds given the terroir that they're actually living under. So I can do the same thing, and I can do it so you can see it a little bit better. But you can see the red boxes and blue boxes. You can see the groupings that occur within the system. And by the way, what you're looking at is rapid fingerprints of these individual isolates as analyzed. And again, the same kind of learning that we learned last time. Okay, then moving now to the independent of culture. So this is using TRFLP. Here's the analysis. We did some clustering of this work. But you can actually see, so we've changed the TRFLP to an image looking like a gel. You can see here that coated in yellow are the actual hens, and in actual day of hatch are the blue. And you can see almost perfectly yellow separates from blue. And if yellow separates from blue, you're not getting mom's microbiota. The maternal line microbiota is an important part of that GI tract. So you can see the kind of names we're missing. Enterococci is very common. Johnsonii only in the hens. Salvarius only in the hens. Crispatus very dominant in the hens, but not in the day of hatch. Casei was common throughout, and Pedococci was kind of hit and miss within the day of hatch. When we did this across all the companies, it's the consistent numbers and names that keep coming back and teaching us. This was complex too. Remember, we have nearly almost no amplification. Only when we go far beyond do we actually get Lactobacillus casei, and that's the only thing we get. I guess we got maybe 1 or 2 enterococci. So this was, again, day of hatch, completely segregated away from maternal line. So now moving to phase 2, we already began to see things that were missing in the normal microbiota at the day of hatch. So we said these are our candidate strains. We want to see what happens when we take normal microbiota from the hen and put it within the day of hatch bird. So we began to look at individual isolates that were common, the core microbiome that we began to see. And here's one of the isolates. So this is Ellen Davis's work within our team, looking at the function. In this case, we're looking at expression, MIP2, TNF-alpha, and IL-6. You can see here the regular LPS challenge model in which we get the fold expression of the actual genes represented there. Here's a control strain, which is actually looking at the strain itself and in the presence of the LPS. So you can see 1E1 was a strain that was very important because it bound to toll-like receptor 4 and 9 and was important in the quiescence of the system. So this actually is one of our controls. Oh, the new strain that we're looking at is 1037, and you can see on its own it's got an inflammatory response, but in the presence of LPS, it quiets that inflammatory response quite nicely. So functionally, this is a strain that would help in early inflammatory challenges within a bird. I'm going to jump to the end point. So this is when you have to grab or lasso that star, and I'm going to use that picture. That's a great story. When you lasso that star and put 1037 into the system, what do you actually see? In this case, we put it in with a little bit of bacillus. The bacillus are there to help control that avian pathogenic E. coli. That's a direct inhibition based on the antimicrobials produced in the bacillus. We actually ran this 1.5 million birds for 12 weeks, 6 on, 6 off. We captured the microbiota and also, I'm going to show that, but also looking at some of the gut function or the gut microbiology as well. So turning now to the gut microbiology, looking at the day of hatch between control and treated, and 7-day, we had a significant reduction actually in the avian pathogenic E. coli at 7 days. When we looked at what that meant in terms of the histogram, you could see, in fact, we had more guts that were cleaner, binned with less. That's the log number of the avian pathogenic E. coli. When we looked at the actual performance of the birds, here's what we saw. a decrease in 7-day mortality, and in this particular customer's case, and just what Julie was talking about this morning in mass shops, oftentimes in an antibiotic-free environment, you see huge variability. Well, one of the things that a lot of producers want is to actually reduce that variability. In this case, we saw a reduction of the coefficient variation, which was an improvement. Net return for this producer on the birds that they actually harvested out of the system was a 4-to-1 return. So we were, in fact, right on. Today, they're commercially using this system. Okay, now we turn to the swine. We've got 4 minutes and 30 seconds. I'm good. All right, so here we are, phase 1, basically the same thing. But we have to do a lot of background work. We have to look at that variation across a GI section. And thank you for the answer. The GI section is actually quite critical. We've seen the same. Particularly, what sections you look at, you'll actually see a lot of difference. We're going to try and build those sections back together, however, and say this is the average of the walking pig. And when you do the analysis, and say if I take an average chunk of the gastrointestinal tract and combine those and pool it, you actually lose the integrity of the actual section. So it actually has to be done by taking section by section and pooling the data in silico rather than pooling the sample. We're also going to look at variation across pigs, across cell flow, which could be independent or could be confounded by site, and then also going to look at company. Because in the end, is the core microbiome of one swine company the same as the other? And early data would indicate probably not. Once we identify the variation, then we're going to start to use some of those samples and begin to understand those key microbial isolates with that core microbiome. Microbiome meaning we're going to go down to that genome understanding, that metagenomic analysis that we want to get to. Understand those functions that we've done before, and determine, of course, that they are potentially important in improving performance and health of those pigs. So where we are today is right up here. I'll talk a little bit about the samples and what we did. The parasesophageal region, if you're not familiar with that, is the first part of the colonizing area for lactic acid bacteria as well as others. It's an important part because it kind of is the beginning of how and where the bacteria are going to colonize. In addition, we're going to take jejunal sections and ileal sections, and then we did enumerations, of which there wasn't a lot of excitement, so I don't think I showed you any of that data, and then TRFLP analysis to kind of bring it home and understand where we are. Okay, so unlike a bird, however, we have a very advantaged system in which the baby pigs are in constant contact with the sow. So, in fact, our prior work had shown that we could actually feed a bacillus to the sow and we could get it within the gastrointestinal tract of the baby pig through just environmental contamination. So, this is a good day. However, the bad day is that pig is going to be weaned from that sow, which is going to cause a huge change and stress on that pig, both socially as well as nutrient changes that are going to occur to make a huge disruption within that microbiota. So our prediction was, don't worry so much about this time. There's going to be normal succession events here, but there'll be a catastrophic or a big event at that day of weaning. And so we'll show you some of that data now. So, starting first with 16S sequence of the actual isolates that we got through the plating systems. So that's the normal cultural data. This is at the high level, so this is 16S taxonomic identification at the level of the family. And you can see the family names here, and this is the case across ages. So we have pre-weaning, the day of weaning, 3 days post-weaning, and then post-weaning as well. There's a beginning change you see at the day of weaning, and the issue is that some of the pigs were on the truck already, so you already had a changing microbiota. When we drill down and look at the details of the 16S, you can see already that it gets quite complex. But again, the pattern pre-weaning is pretty much conserved, and this is, by the way, the average of 5 sites, 5 pigs per site at each day, and across these 6 time days that we've captured here. So you can begin to see at the genus level the kind of organisms we're seeing, and a big shift that's occurring within weaning and post-weaning that's normal part of the succession process. When we use TRFLP and look down, we always want the cultural data to kind of back us up, because in the end, the cultural data is going to tell us, in terms of our picking, where we need to go based on that independent culturing method. So here we are, looking at TRFLP. Now we're using primers to actually get the total bacteria, These are the averages across site. It's not that critical because site doesn't have as much importance as actual time. So I'll switch here. Look at lactic acid bacteria by site. You can see there are differences across site for sure. More importantly, when we look at now the cumulative of the sites across time, again, for total bacteria, some complexity, but I want to break it down, and you can start to see a huge difference between day 20 and then post-weaning day 23. So these are the rearrangements that's occurring. Now, the data that we get from the producer is critical in this process because that producer can help us understand high-performing, low-performing. And now we can actually drill into the data, develop correlation models that begin to set up that future, which we'll have to prove by causation, but we start with correlations first. And so here's an example of the best performing site by age, and you can see the huge change that occurs between pre-weaning and post-weaning. So target organisms for future DFMs would come from an understanding of the post-weaning, in this case, happy pigs that are performing well, compared to sites, other sites in that system. And the better the models, the more black and white you have it, the better off you can see the differences between what might be that star that I want to lasso. So here's where we are today. We understand the diversity. We know that it is by section and by pig and by site. So you have the complexity, but you have to be able to pull that complexity together to understand how then we can drill down. And drilling down to TRFLP tells us which samples that we want to actually drill down deeper and actually look at deep sequencing to understand strains within species. And then how we're going to move forward from here is that the correlation, we'll begin to develop those correlation models between the isolates and performance to begin to understand, using multivariate statistical analysis, to begin to understand which ones are correlated to performance and could be a candidate DFM. That's correlation. Until you feed it and prove it and understand its function, which is the next steps for us, do we actually get a causation step. So we're a good 6 months. Rob, you okay with that one? 6 months away. He's not. He's good. So 6 months to a year away, but this is the process that we go through. It's been successful in poultry, and here's where we are today in swine.